Hello, my name is Brooklyn, and I did this project with Ariana, Megan, and Lily, and the purpose of our project is to explain in depth the creation of protein chains, also called polypeptides, and the process of protein synthesis. Gene expression is a process in which DNA is converted into a functional product like a protein to show that gene's qualities. There are two main steps of gene expression: transcription and translation. Transcription makes a copy of DNA that can leave the nucleus and starts by being split by helicase, and a new strand of mobile DNA called messenger RNA. The mRNA molecule is elongated by RNA polymerase 2, and once the strand is completely synthesized, transcription is terminated, and splicing and RNA editing occurs. After transcription is over, translation occurs. Translation is reading that mRNA sequence to build a polypeptide or protein. Initiation is the beginning of translation. It is where the ribosome meets with the mRNA and tRNA and translation starts. Next is elongation, which is where amino acids come to the ribosome by tRNA and link together to form a chain. The last step is termination, which is where the codons on mRNA for stop is read and the polypeptide is done being coded for and is released into the cell's cytoplasm to do its job. The role of DNA is to code for the mRNA sequence, which initially communicates the sequence of amino acid bases needed to create a protein. This mRNA has codons that attract complementary anticodons from the tRNA that, after connecting, release an amino acid connected to the other end of the tRNA to the ribosome. This process is repeated until all of the codons on the mRNA have been matched with anticodons from tRNAs, and it creates the protein chain the DNA initially coded for. The structure of a ribosome is made up of rRNA and proteins and is essential to gene expression because it holds the mRNA in the ribosome while it is being read by the ribosome and tRNA until it is finished making an amino acid chain. It also holds the polypeptide being created with the amino acid released from the tRNA together while it is connected to the mRNA codon sequence. The ribosome also contains enzymes essential to protein synthesis. The parts of the ribosome are the small subunit, the large subunit, the A site, the P site, and the E site. The A site is the attachment point, the P site is where the amino acids are dropped off, and the E site is where the tRNA exits after it drops off its amino acid. We will now demonstrate the process of protein synthesis within the ribosome. So first, the mRNA enters the ribosome, and the codon sequence is read. Then tRNA goes to the ribosome and picks up an amino acid that matches the codon of the mRNA. Then the tRNA moves to the E site and leaves. The next tRNA that codes for a different amino acid picks up its amino acid and drops it off. And then it moves to the E site and exits the ribosome. Then the last tRNA drops off its amino acid and moves to the exit site and then leaves. Okay. But how does all of this work? First, there is an initiation, which at the P site, the first codon of mRNA is the AUG codon. The AUG codon is the starting codon, so the rest of the protein sequence follows behind it. Then this leads to elongation, where the tRNA meets at the A site and moves through the A site to the P site and begins to attach and make polypeptides. Attachment of amino acids only happens at the P site, however. Once that is finished, the now empty tRNA exit at the E site as the mRNA continues to be read by the ribosome. for its encoded information. Lastly, the final step is termination, which, like the name suggests, the stop codon is read on the mRNA, which stops the process of making proteins, and no more tRNA is brought to the ribosome, so the polypeptide is then released into the cytoplasm. Also, fun fact, there are 21 different amino acids that could possibly be coded on the mRNA.